Good afternoon, ladies and gentlemen, and welcome to your SETI Institute Colloquium series. My name is Frank Marchis. I will be the host for today's colloquium. It's a great honor to introduce my colleague, Margaret. She's going to be our speaker today. So we spend a lot of time together, and it's always interesting to present someone I see every day or who I talk to on a daily basis by email and know every tiny secret about your life now. It's great. So Margaret is an ecologist at the SETI Institute who works with NASA on astrobiology and planetary protection. She's currently working as the lead editor of a workshop report on planetary protection and Mars missions. Over the past 2 decades, she has served on numerous US and international studies on avoiding forward and back contamination on both robotic and human mission in the solar system. In addition, she's actively involved in the analysis of societal, ethical, and policy implications of space exploration and the possible discovery of extraterrestrial life. I think that's why you work here, right? Margaret has been involved for decades in science education and outreach about astrobiology through the mass media, school, museum, and public presentations. So Margaret received BA degrees in biology and master degrees in energy management and policy from the University of Pennsylvania and a PhD in ecology at the University of California at Berkeley. So prior to work to be at SETI Institute, she was a professor and researcher at Stanford University, assistant dean at UC Berkeley, and senior science policy analyst at University of California, Office of the President. So she has a very interesting career. As you can see, she started out as a marine biologist, studying the environmental impacts of nuclear and fossil fuel power plants. Later, she did some research on mud snails that were brought to San Francisco Bay on the transcontinental railroad. So it's a very interesting path, and does bring her, definitely it's logical, to the study of planetary protection and the design, how we could design mission to explore Mars with humans. So please let's join me in welcoming Margaret.
So this is a different type of seminar than most of you are used to. I know you come here and you listen to extrasolar planet talks or SETI talks, and this one is really different. I'm bringing you back home into the solar system, and I'm bringing you up against things that are science fiction or popular literature, or even in the bottom, Mars One, a proposal to send humans to Mars one way. So there's all sorts of things, and NASA has thought about what we would do for a human mission, and I thought it would be good for you to realize just how much long-term planning there goes into something like this. And even though we're not likely to know how the mission would be or who will have the first mission, we know that planetary protection will be a part of it. So I'm going to give you a talk on planetary protection, and since I'm guessing that many of you astronomers, astrophysicists, cosmologists, and technical people probably know, you think about this as the government coming in to tell you what to do. We're here to help you and regulate planetary protection. Well, I'm going to tell you more than you ever wanted to know about it. And also, we're building on our experiences both robotic missions and human missions in the solar system. And before you start planning a trip, it's a good idea to know what your trip is all about, and that means what are the travel details? Are you going to work? Are you going on vacation? Or are you going to the moon or beyond? So you need to know that. You need to know the risks and requirements that go along with it. You need to think about who you're going to integrate your information with, because biologists don't just go to Mars by themselves. They need technologists and others, engineers. And how do you plan it so that it really happens? So I'm going to bring you into what's going on now within NASA, and has been for well more than a decade, and that's the development of requirements. What would it take to have planetary protection requirements for humans to go to Mars? And we're in the process of developing not just the idea of preventing contamination, but what level of contamination makes sense. If you think about it, you have, thou shalt have clean water. What's that mean? Someone needs to take the science and say swimmable, fishable, drinkable. Each one of those have different standards and criteria, hopefully set based on science. So that's where we are right now. And NASA does a lot of its work, and the international agencies, by looking at workshops. And they set up workshops and invite people together and say, what do we need to know? And that's what I'll tell you about today. That's where we're going with this. And we're identifying what are the research areas we need to be looking at and the technology that needs to be developed in order to then set ourselves on the way to Mars. So first, planetary protection. What is it and why should you care? Well, believe it or not, planetary protection goes way back to the beginning of the space age. And biologists, even before Sputnik was launched, suggested there was a need to think about sending microbes on spaceships that might go to the moon or beyond, because we were, after all, going to look for life out there. And the last thing you want to do is bring life with you. It's a contaminant. And so they were asking, how do we keep our science clean? And the Outer Space Treaty, right from the beginning, when it was written and ratified in 1967, had Article 9, which said, avoid harmful cross-contamination of planets and adverse effects on Earth from space exploration. That translates into two things. Avoid forward contamination. You want to preserve planetary conditions up there. You don't want to go and look at the moon all messed up. You want to go and study it the way it is in a pristine state. So you want to be able to say, if there's any life up there, you want to recognize it as biological life that might be on the moon. So you want to make sure you don't send organic or biological things with you. So that means avoiding the introduction of terrestrial microbes on spacecraft that are going from Earth. Then we also care about this planet. So when you bring something back and you want to study it in a lab, you want to worry about protecting the Earth and its inhabitants. And we know much more now. It isn't just public health. We're thinking about the whole planet as an ecosystem and how it works. So we want to avoid anything that would be potentially hazardous. And that means not just a toxic something, but more likely a replicating organism. If it's toxic, you can deal with it in a laboratory. But if it's replicating and it gets out, you've got problems. So avoid bringing things back in your samples or in your spacecraft that would be potentially bringing back something uncontained. So this applies also, which surprises a lot of people. It applies not just to the governments. Over 100 countries have signed the Outer Space Treaty. It applies also to non-governmental entities. So although they may not recognize it or won't want to admit it, when a space company wants to plan going beyond Earth orbit, they do need to think about this. These are international United Nations regulations. So how do you plan a mission with planetary protection? So you're taking these treaties and policies and turning them into something that really amounts to responsible exploration and good science. You want to make sure that when you go to do your science, that's really what you're doing. So the National Academies of Science here and elsewhere have said, take a conservative approach. If you don't know if there's life up there, assume it's so, and that anything you bring back could have life in it. And that's, so really you're starting to think about invasive species, pandemics, and the things we think about here on Earth. And not surprisingly, things changed since the time the treaty was passed. So CoastBar, the Committee on Space Research at the bottom, is an international organization of scientists who keeps updating the treaty standards, criteria, and whatnot. So planetary protection in a nutshell is responsible exploration. Just avoid the past mistakes. We know that a lot of good things came out of early exploration, but some things weren't so good. So with planetary protection, every time you plan a mission, all the planets, all the time, you have to think about planetary protection. And it's kind of like environmental impact assessments for the solar system. You're avoiding exotic species, you're minimizing hitchhikers, and you're keeping your science pristine. So we have a lot of experiences with planetary protection. It's not just something that was written down and then we move on. We especially have them in robotic missions. So especially what we've seen on Mars, from the Viking missions in the 1970s on through Pathfinder, launched in '96, the Spirit and Opportunity on through Phoenix, and now Curiosity. Planetary protection implementation has been a part of it, pre-launch, to protect the science and to protect the planet. And it applies to landers, orbiters, drillers, anything that goes there. Basically, this is a nutshell of the CoastBar policies. These are the international policies. Basically, it doesn't mean that every time you go any place you have to worry about planetary protection. If you're launching something to the sun, it doesn't matter what's biological, it'll get cooked. You don't have to worry about it. So if you're not going to a place that is of direct interest for the understanding of chemical evolution, again, it's that link to life and where did it come from, then there's no protections. But you still do have to apply to the Planetary Protection Office of your appropriate country and tell them what you're going to do. And all of this is documented and given back to the United Nations. So CoastBar is a non-governmental organization that reports to the UN Committee on the Peaceful Uses of Outer Space. So they've got information on where all of these missions have gone, where they've crashed, where they've landed, what they've done. Others, significant interest, but only a remote chance that contamination would be a problem. Think of Venus, say, or Pluto, for that matter. We don't think there's really a lot of life there on Pluto or even Jupiter, but then we started finding Jupiter and Saturn have some very interesting moons. So maybe the moons are there. So it's celestial bodies, that not just planets, that we're concerned about. But if you get to category C, of significant interest, because they have potentially habitable landscapes, places like Mars, places like Enceladus or Europa that have water and ices, these are places where you could have significant contamination. So you want to be really careful. Whether you're flying by it, whether you're orbiting, whether you're landing, whether you're drilling, all of those things take place. And the bottom one, any solar system body, doesn't matter where you go, if you're coming back to Earth, you have to think about what you've done and make sure that you're approved to come back. Now, if you're coming back from Venus or the sun or someplace that isn't habitable, you have an unrestricted return. Come on back. If you come back from a place like Mars or someplace that could have liquid water or conditions where life might exist, then you are a restricted return, and those restrictions are quite severe. So basically, planetary protection, when we're doing it, involves cleaning things, bioburden reduction, that just means the biologicals, the contaminants, and the biology, the microbes, and assaying to prove that you did meet the contamination controls that were set. This is the picture on the far left is actually the Viking life detection mission and the instrument that was sent up there. Now, if you look at those guys, mostly guys, there's an awful lot of skin showing and breathing faces. So we didn't have the same view in the 70s about what microbes were all about. We did, however, the middle picture is the Viking, they call it the Viking Casserole, the entire spacecraft, not just the parts that were going to touch Mars and do the examination for life. The entire spacecraft was put in a room-size oven and baked for a couple of days at a temperature high enough to presumably reduce any kind of microbiological load. In addition, you have the entire transit time to the planet, so you've got extensive radiation hitting. So there's lots of places where we can take that bioburden reduction. And the far right one is assaying the Pathfinder rover. It was a small thing about the size of a microwave oven. And when that one was assayed, it too did a really good job up there on Mars. So now what we have is a lot more complicated. So we're still using culture methods, which is very interesting. So how do you move from the old culture methods where you swab something, think of the swabs that are used for your strep throat or something, and they put it on a culture and they grow it up. Well, now we know enough about the human microbiome, or any kind, to know that probably 95 to 99% of microbes are unculturable. You don't know what to feed them to make them grow. So what they do is heat shock things, figuring that most organisms will die at a certain level of heat. And anything left is in spores, and so the standards for these are set in spores. Spores per area or spores per whole rover or spacecraft. And when they go out now, whether it's the small microwave-size rover that went up on Pathfinder or the 1-ton Curiosity rover that went up, the limit of the spores is about 300,000 spores for the whole spacecraft. And so they take care of lots of things. And these are photographs of people actually swabbing and culturing. They're brought back to the lab, the woman down in the bottom right, and there's the whole rover is not the only thing that is cultured. So the far right middle picture is actually the parachute. So we think about the parachutes and other things that go. So before the launch occurs, here's the payload fairing with the MSL package on the inside. All of those things are signed off on to indicate that all of those assays found the requisite number of spores. And they did close to 50,000 assays. These are, think about 50,000 cultures in a lab, a blood lab or a strep lab or something. So a lot gets done before they're launched. And so this is what we do for robotics. We have a really good experience record. Okay, then we've got things that we know we've done a lot. So something like the Mars Reconnaissance Orbiter can take pictures, and we know that planetary protection isn't just because of the science we want to do. Things can crash. They can go wrong. So we can actually see where a lot of these things landed. And so when a heat shield comes off, or a rover lands, or a parachute comes down, those have to be cleaned too because they're landing on the surface. And and we also say, well, there could be life up there, so you wouldn't want some of these things to land on something. And just so, to give you an idea of how current it is, no matter how long it's been around, planetary protection has relevance even today. This was this week, and I first saw it in Fox News and the Daily Mail from Britain. They discovered there was a paper that said resurrecting monster viruses from permafrost. It was a study that was done in Russia, and they drilled down, and what they found was 30,000-year-old bugs, viruses, 30 meters deep, and they reanimated them. Basically, they brought them into a BSL, Biosafety Lab 4. Those are the moon suit labs, the glove box labs. And they said, can we infect amoeba? And they could. Now, the interesting thing, so then the experts are starting to say, well, this is like climate change. You're talking about going up there. We've got melting permafrost. We've got oil and gas exploration going down into the subsurface. So there's even people who have said there's people up there that who had smallpox. You can resurrect smallpox. So all of this now, so they've put this out as climate change and monster viruses. But really what it is a paper, a legitimate science paper that was in the Proceedings of the National Academy of Sciences. And it was the French group of people, Clavelier et al. And what they found is 4 different types of giant viruses with different structures, different sizes, genome length, and replication styles. And this one they just found, Sibericum, has more than 500 genes. And another one that they found in 2003 has 2,500 genes. In comparison, influenza A has 8. We don't know that quantity necessarily goes along with contagion, but we sure wouldn't want to be putting that stuff out. So there's the planetary protection piece. So we think about it even here on Earth. It's not that the risk is enormous, but the risk is not zero. And so if we want to do responsible exploration, we think about that. We also thought about it when we were doing sample return planning. That's when I first got involved in this. I had done invasive species mud snails in San Francisco Bay that came in by the Transcontinental Railroad, an emerging technology. And so when we talk about going to Mars and bringing back samples, it's the same kind of thing, an invasive species or genetic engineering, something made in the lab and put out into the field. So we did a whole series of workshops that were planned in 1999. We were talking about launches in 2005 and a return in 2007. All of these workshops were held and came up with a draft test protocol. What would you do if you brought back samples from Mars? Just rocks, mind you. Rocks, pebbles, dusts, gases. Could there be life in it? And if there were, where would you put it on Earth when you bring it back? And what kind of test would you do? That's all that's going on. So what resulted from these was an entire protocol, again being done in biosafety labs. So we worked with Center for Disease Control and folks over in Europe as well. And so there was a rigorous protocol. Contain it, keep it contained, process it, and know what's there scientifically. Look at it and put aside some of the samples so that you'll have them for future generations to look at. Do some more life detection tests. Is there anything you recognize? And even if you don't see anything, do biohazard tests. So what is a biohazard test for something from outer space? Well, You have to start thinking about it. So anyway, it was all done. It was all planned to be done. We didn't go yet to get samples from Mars, but we've thought about it, and we have a lot of experience. Now come back to experiences for humans in planetary protection. We obviously have tremendous amount of expertise and experiences with human missions in orbital space, in orbital space, lunar missions, International Space Station. But come back again to the Outer Space Treaty. If you notice, most of the legal issues that have to do with anything are in low Earth orbit, ISS, suborbital, and geostationary. Have to do with satellites and orbits and frequencies and things like that. So there's no planetary protection when we send up humans in space, because we know we got microbes. We know we're in Earth orbit. Contrast the moon and other celestial bodies, which have only been explored by scientists and government agencies. Those are the places where planetary protection holds. And all the other issues are TBD. Environmental management, what's an appropriate level of containment, who gets access to it. All of those questions are something that the lawyers will have a good time with before long. But the important thing is planetary protection has only been done on robotic missions, except the one important time that we all know about, which is the Apollo mission. We have gone there. But it turns out it was a very limited time. And when we went there only during the Apollo program. We were there for only Apollo 11 to 17, and remember 13 didn't land. And it turns out that we know enough to know now, because we contain things very well. We put the astronauts, we put the samples in containment labs. Actually, when they landed on the ship, they were in a containment lab. They stayed in it until they were taken off the ship, and they were flown in a cargo plane to Houston, where they stayed in containment in a quarantine lab for a full month. Now, if there were life on the moon, we would have it here on Earth, because we know now that what we did in the early 70s was not sufficient. So where we're at right now is that the laws have changed. EPA didn't even exist at the time Apollo 11 happened. The science has changed clearly. The technology has changed. And when you look at something on the moon, this picture on the moon is, I believe, Apollo 14, and all of that aura around, that's the venting of the spacesuit. So when we vent, we're putting out microbes. We're putting out all sorts of organic contaminants. So is that amount of venting okay on Mars for the length of time that people have to stay there and for the number of people that are out there? I don't know. That's what we have to think about. We also are moving from designs that have been on orbit for a very, very long time. And if you think about the astronauts in orbit, they're in nice white spacesuits. When you think about the astronauts on the moon, there was a lot of dirt. It was lunar regolith. And some astronauts have said that if they stayed more than 2 weeks, the zippers, the suits, the seals would not have worked. Think about going to a beach. You got Velcro in your zipper. I mean, you've got sand in your zipper or sand in your Velcro. It doesn't work anymore. So what we're saying is, we only had limited planetary protection with humans, but lots of human experience. And we have lots of planetary protection, but not on human missions, only limited on human missions. How do you put them together? And that's what we want to do. So how can we feed forward in thinking about it? Can we build on what we have with space station, with going back to the moon, with asteroids, and any other place, including Earth analogs? So this is where we are. What's the trip? Think about a trip and the travel details first. This one boggles my mind. Okay, when we went with the Apollo missions in the late 60s and early 70s, there were only 6 landed missions. Moon is 250,000 miles away, and a round-trip mission was 1 to 2 weeks. And we were Earth-reliant. Even on International Space Station, we're Earth-reliant. If something goes wrong, you come back to Earth. If you have waste to dispose of, you put it in the shuttle bay cargo area, and bring it back to Earth. So everybody is definitely working off of Earth. Now, and even in space station, it's very much that way. It's been occupied since November of 2000, It's in low Earth orbit. We don't have to worry about planetary protection, but we can use it for a lot of research. It's the size of a 6-bedroom house, so it's pretty comfy for the astronauts. But it only takes about 6 people at a time. And as I said, it's only a couple hundred miles up, and it's got a full 1 acre of solar arrays. That's compared to a football field there. So it's unlikely we're going to launch something like that to Mars real easily, so we have to start thinking about it. Also, when you think about going to Mars, it's a different environment when you get there. That's Mars. No infrastructure. And you're going to have to, of course, think about, so we got lots of ideas and plans, but what do you do to build that infrastructure? And what does it take? And so most of our missions think about protecting the crew and having things safe when they're on orbit and having the technology to run it. But protecting Mars and protecting Earth are these 2 things that they've never had to worry about before. So we're adding that in to experts who already know how to do human missions. So in the beginning, there was a little bit of a tension. Excuse me, I know how to do human missions, and the other, excuse me, I know how to do planetary protection. So how do you get these guys together? So for the space station, it's 250 miles up. The missions last at most about a year. For the moon, 250,000 miles away, round trip 1 to 2 weeks. For Mars, 50 million miles just one way. Round trip will be 2 to 3 years. So NASA's been doing a lot of different mission profiles, design reference architectures, as they call them. The idea is to send up things first, launch cargo, let it be, build the infrastructure that you need up there ahead of time, so that when the astronauts come, they're not just staying in that small spacecraft that they flew there on. They're getting out into a prepared infrastructure. And then they're staying on planet for 500 days or so. If you look at it, you guys probably all know this, but you know, Earth goes around the sun in 12 months, and here's 12 months, and that's where Mars is. So you got to wait, and your launch opportunities are only every 26 months. And what are you going to do when you get there? You're up there for 500 days. Let's hope that they're going to be doing something useful, some science, some exploration. And so we've got to think about it. What is your mode of transportation? What are you going to do when you get there? How are you going to feed yourself? All of those things. So it's really fun to go into classrooms and deal with young students. I can even say, what did you bring to school today? Where's your lunch? You know, how long are you going to stay here? How are you going to get there? And so you make them go through the same thinking and realize that what we've got is very similar to long-range planning. So these are the kind of risks we have. Pascal Lee, one of our scientists, has written a wonderful book, Mission Mars, and he's doing the same thing. It's written at 5th grade level. But basically, the risks and requirements are exactly what we think about when we're planning human missions. How are you going to get there and return? How many crew members would be okay to send? How are you going to build your shelters before you arrive? Where will you get your water, your food, your supplies? And think about it. Well, you just melt the water. Well, if there might be life in it and you melt it on Mars, you got to drink it. Another thing with the sample return from Mars, we ask the question, if you have Mars soil, Mars regolith, and you put in Earth seeds, do you end up with a hybrid biology? Low probability, but you can't rule it out, so you need to look at it. And on about spacesuits and rovers and moving around and communication. You have that delay time for communication because you're at least 18 to 30 minutes away. What other hazards do you have to deal with in space? It's radiation, it's the pathophysiology of muscles and bone, and now they're saying a lot more the fluids moving up into the head, so you've got eye problems. So there's lots of things we have to work out. And how do you do the psychology? Once you leave Earth orbit, when you're about 2 weeks away from Earth on the way to Mars, Earth will look like that. It won't even be a pale blue dot. It'll be a dot. So you're in the black of space. And what does that do psychologically and behaviorally? And imagine when all of us get in an airplane and we're going to the East Coast, and after 5 hours, you're glad to get out and have some real food. And well, they do give you the wine, but anyway. So you think they'll be on their way for 9 months. And what about emergencies? So all of those things together mean that when we're planning a mission, NASA is saying, let's take an incremental approach. We don't know which mission will go first. Will it be going back to the moon? Will it be more time on space station? Will it be asteroids and having astronauts live out far away in space, away enough from Earth that they're not Earth dependent, they're not reliant on things, but these become proving grounds. And we can do those proving grounds without having to think about planetary protection. I can go to the moon because we know there's no life and it's one of the lower categories. So we could test things up there and see how far the microbes go. Do they die or not? We can think about an asteroid. Probably not habitable conditions, unlikely to have life. So we can go and do our tests up there. So part of it is thinking about how do you blend together astrobiology science and looking for life and knowing about planets, planetary protection, all the human mission planning that goes into the biomedical side of it, flight operations and what it takes to get where you're going, all the systems that support the astronauts and the space station, biomedical, health, and all of that. So that's where we are right now. We are at a place where the current context is we're saying, how do you take that planetary protection set of constraints, that's an international policy, and say, what does that mean when you're designing a mission for Mars? And so NASA has planned a planetary protection and humans workshop. And what happened is 2008, CoastBar, after a series of earlier workshops in 2000 to 2005, where we got people together, we thought we would be planning for missions to Mars. We got the engineers and others and said, what about suits? What about food? What about? And so we had a series of workshops and got together, and that resulted in a set of international policies that were guidelines and principles. So that wasn't numerical, and it just said you are still going to have to worry about planetary protection when you go to Mars, even with humans, and we'll have to develop the details later on. And so when NASA got together in 2012 with its Planetary Protection Subcommittee of the NASA Advisory Committee, the NAC, they recommended the need to take the CoastBar requirements, the international ones, and turn those into controls and requirements for how NASA should do its missions. Each country does it a little bit different, but they're all living up to the same policy. So the Human Resources Group and the Planetary Protection Group started to get together. And in 2013, they realized, we just don't have enough information yet. And so they said, we have a lot of knowledge gaps. Before we can start telling you, this is the vent size, or this is what we have to do with spacesuits, or this is what we should do for quarantine up there, we need to start to address some of the knowledge gaps. And they adopted an incremental approach. So rather than doing an attempt to go right for requirements, they went for NASA procedural instructions. Let's set up a whole series of things that will move us towards requirements, but not get us there right away. Now, the important thing is these. The CoastBar policy in 2008 is what's holding. And they set up principles for human missions to Mars. Safeguarding the Earth is the highest planetary protection priority. And they're even talking about, if an astronaut's contaminated, do you bring them home? That's not different than what was done during the Apollo missions. The commander of the USS Hornet, which was the ship that picked up Apollo 11, had two sets of orders. And he said, one said, if there's contamination, then the ship should stay at sea, and that will be the containment vessel. And if there's not, then we'll come to Pearl Harbor and offload the quarantine vessel and send it on an air cargo plane. There was also the interesting thing of the president was on that, remember? Nixon was, he landed and welcomed the astronauts. So what do you do if that happens? So planetary protection will still be important when we go to Mars. And we know that if we're going to have human-associated contamination up there, it's going to mess up your science, so avoid that. The third one, for a landed mission, we acknowledge that it will not be possible for all human processes to be conducted within entirely closed systems. As much as you might like it, you're going to have to vent. Okay? It's like when you go down in a deep submersible. You may want to reach out and get that giant clam or that 6-foot tube worm, but you can't do it. You have to do it with a robotic arm. So it doesn't mean you'll stop doing the research, you'll do it just a different way, more autonomous, a robotically autonomous way. And the fine thing, the crew members exploring Mars will be exposed to Martian material. So what kind of information do we need to know about that? They can't pop a pill and get better or take an antibiotic. So all of these are the guiding principles that we know about. Then, based on those, we said, here are the guidelines that we'll use for implementation. Continuous monitoring and evaluation of all the microbes up there, terrestrial microbes. A quarantine capability should be built in for one person or the whole crew. We need to have a protocol set, just as we did for the Mars sample return labs. Now, we've known enough up there now to say that we need to protect special regions. These are these areas scientifically that could have water ice underneath, could have places where we think the environments are more habitable. So we don't want astronauts going into those. We'd send robots in first. Any pristine samples that would come back have to be brought back as a restricted Earth return, full containment. What does that mean for humans? And an onboard crew member should be designated as a planetary protection officer, basically, to keep sure that the protocols are lived with. And that, again, we take a conservative approach. So it's not like if you pull all the information that we already have together, the context is we need more information, but we already know we can build on all those workshops, all the other planetary protection work, and we know the key systems, the operations, and the equipment that could be impacted by planetary protection and microbes. And everything from the monitoring to decontamination, you'd want to, if you go outside and you come back in, you'd want to clean your equipment or clean your spacesuit. How do you do that? You'd want to think about the spacesuit design. And in fact, at some of these early missions, it was very interesting because the astronauts, the engineers who were there were saying, don't build it like a US spacesuit. If you think about it, they put on the bottoms, they put on the tops, they put on the helmet, they put on the gloves. This one, I remember one engineer said it should be built like a Russian spacesuit. They get into it like a refrigerator door, step inside and close the door. You have one seal. You still have all of your movement, your joints and stuff, but so they've actually done that. And so the planning for Mars missions would have a different kind of spacesuit. So all of this material inventory, you have to think about what you've got there, what could be, how do you maintain tools, how do you keep your lab area separate from your habitation area, how do you make sure that if they're doing some research in the laboratories, stuff doesn't get out into their food. And so the gaps that we have, if you look at the whole mission, we need to know more about the Mars atmosphere and what it's all about, the landing site characteristics, where on Mars would you land? What do you do to keep crew healthy the whole time they're up there? What about dusts? And here's something where engineers think about dust because it can affect their equipment. It could also make biofilms and corrosion. Planetary protection people think about dust because those are the transport mechanisms, if you will, for microbes. And the biomedical people think about dust because they're thinking about what might get into an astronaut's lungs. So instead of having 3 separate groups work on dusts, bring them together. So that's kind of what's going on behind the scenes. Radiation monitoring and effects. Well, radiation is not a good thing for humans, but it may be really great for sterilization. So here's something that's both a positive and a negative. And maybe you can do some of your early missions to Phobos and Deimos. Don't go on to the Mars surface. Go to a lesser restricted location, and you're still close enough to do research with robots and such, and do some of your early testing, and it's easier than being on orbit. So there's different ways to do that. But if you look at the bottom, the knowledge gap includes all of planetary protection, and that's looking at forward and back contamination, what are the biohazards on the Martian surface, what do you do about special regions, and what about microbial survival in Martian conditions? It is possible, for instance, that the microbes could get out and every last one of them dies. That's terrific. There goes planetary protection for outside things. So you can, when you know your information about the microbes, you can use that to mitigate. So mitigation is the key. And so that's where we are. So this NPI, this procedural instruction, is a pathway. And the intention is to Develop capabilities to monitor communities, microbial communities. For every cell in your body, you have 10 microbes. We are just finding out about the human microbiome and humans as an ecosystem. So there's a whole new set of research here that also touches to the same kinds of things that are going on in the health community. So here's SETI Institute sitting there doing the same kinds of things that people in the health community might be looking at. We need to develop the technologies for minimizing or mitigating the contamination release. Cleaning, support systems that minimize things. Think about the advanced life support systems that are up there, the waste containment, the how we do things, where we put things when we're done with it. How do you do washing and cleaning up there? What do you do with your food? So all of those things, the technologies that would mitigate and minimize the contamination. And then think about those environmental processes that contribute to transport, moving the terrestrial contaminants around, or the Martian contaminants. And think about that. Can those outside conditions be used to sterilize some of the contaminants? So we held a workshop that was sponsored in part by SETI Institute, and it was over at NASA Ames. There were 85 attendees in 3 subgroups. One subgroup looked at the microbial and human health areas. Another one looked at the technology and operations. And the other one looked at transportation, dissemination of the microbes. And each group was asked to think about these 5 questions. What are the planetary protection research and technology areas that are critical for solving your problems? What work is already underway? Don't reinvent the wheel. Have we considered enough to include the accidental problems? So you want nominal and off-nominal situations. Make sure we've got them both. Are the existing options for mitigating the contamination adaptable? Can we just bring them over from what we've already got in humans? And are there significant stumbling blocks, aside from funding, of course? So what we wanted to do is identify the specific research needed that could then feed forward as NASA's planning its research budget for the future. And overall, the goals were to gather and discuss that information to move us closer, little by little, to assess the types and levels of research that were underway or that are underway that'll help us, and to build a network of expertise. In part, some of these workshops are pull together the experts and let them interact. And that's some of the most exciting things that comes of it. As a result of some of these, we're now having weekly telecons with people in Houston and other NASA centers talking about different systems. Some people are thinking about, what if I put a rover here and move it around like that? What if the rover is able to just come back in? And what if the rover is our quarantine module? I could put somebody, so what would I want to have in that? So they're thinking in ways that they didn't before. And so we are assessing the state of knowledge and the gaps for all these areas. The first one really looking at microbes and microbe monitoring. The second one looking at technology and operations. And that, frankly, is a really big one because the human people have not had to think about planetary protection. And the last one is a lot of science, the environmental features that can help us avoid contamination. So where we are right now, when I'm not preparing for seminars, I'm working on the workshop report, which also included a very large literature review, pulling together everything from Apollo forward, and then using that as information. Here's where we are. This is what we know. How do we go forward? The workshop has been done, and I'm in the process of putting together the technical report that will be published under a NASA series. We'll turn it also into 3 separate journal articles, one on the workshop itself, another one on this whole literature review, and a third one on a systems engineering approach, because we as biologists and scientists think about this like the astrobiology approach. The engineers think about it more in terms of their technology and how do they think about what fits into the systems. They just say, give me the number. I want to know the size of the vent hole. I want to know my filter. And they'll be happy if you could just give them a number. We take a long time to come up with a number for them. So together we're working. So as we're looking ahead, we've got to move from that NPI process and get to some funding for research projects. And we've already, some of this workshop has already resulted in some interesting findings. One group, an engineering group, on these telecons was saying, what are the microbes inside and outside of International Space Station? Could we swab and sample in the vents and outside the vents? Do we know what gets out there? Well, the scientists have been asking that question for a long time, but what do you think? They send up the hardiest, most extremophile organisms and say, can it live up there? We're saying, what about the human stuff? We didn't bother looking at that. So now we said, great, just have the astronauts swab when they go out on some other test walk outside for some reason. But the microbiologist says, wait a minute, if you're not careful in how you do that, you'll find microbes. And now you'll say they're out there. It could be because he wiped it against his glove, which of course has microbes on it. So we had to first develop a prototype tool. What is the tool and what is the method that an astronaut would use going out, sampling, and then where do you sample on the inside? Is it the bathroom area? Is it the sleeping area, the food area? So make sure you've got the protocol done right. And so already we're looking ahead, and it looks like in November, so the prototype tool has been developed. And in November, they're going to piggyback on some spacesuit testing. And they'll actually do some inside-outside tests. And we don't know what the answers will be. Maybe they'll all be dead, or maybe a few will survive. And it'll be interesting to know which ones survive. And eventually we'll be able to do more of it. And then if we went to the moon or to an asteroid, we could do similar things. We also have done some work, again, Pascal Lee, somebody here from NASA, from the SETI Institute, has done work up in the Arctic at the Houghton Mars crater. And he's looked at when they took a Humvee that was outfitted to go across the ice up there. They took the Humvee that was modified, and they sampled it before they left the Bay Area. They drove it up, and they shipped it for a ways on a cargo plane, and then they drove it across pristine Arctic ice. And they sampled inside the Humvee, outside, in front of, and back of as they went. And they also said before they built the habitat that they've got there, let's see what's up there. So they looked at the area, sampled, and then built the habitats, and now have researchers coming and going, so we have a historical set of data about what's up there. And if things die off or don't go more than 10 or 20 meters away from your site, where you've got your infrastructure, then that's probably pretty good. If an Earth organism doesn't make it here on Earth, chances are it might not have up there. So you can see we're starting, in some ways, from scratch, but we're doing pretty well. And we're also then in the process of shifting, and this has taken years, shifting from culture-based methods to molecular-based methods. And it sounds like, well, why don't you just do it? Well, because what you find is genes and DNA sequences, and not necessarily a representation of what the organism is. So we really don't just want the collection of genes. We want something that, and also that is based as a standard. So you can say this is how many or this type before you launch. So where we are right now, again, is integrating it all, hoping that we'll have some research as we go forward, and that will start to inform us and pass along information to the engineering community. And it will be a very long-term project. And looking ahead even further, oops, sorry, wrong one. What we have next is looking beyond the science. Everything I've told you about has been science exploration missions. And as we look forward, now we've got all these commercial groups that are coming in. They want to do asteroid collections. They want to go and have space hotels. They want to have trips to Mars or one-way trips to Mars. And so how do we make sure that as we develop this material, it also is fed forward to them in ways that are useful and helpful and not something that is a showstopper? Because Planetary Protection doesn't intend to be that. So I'd be happy to take your questions. This is this is one of these things that doesn't have the ready answers, and I've supported my hypothesis or not. This is one that is just a rolling process, and it takes a long time, and a lot of different people are involved, and people here at SETI Institute have been involved.
Given that a lot of the robotic missions that have already gone to Mars have some level of protection, but maybe not a complete level, what's the likelihood we've already contaminated Mars?
That's the same. I can give you an answer. We have contaminated Mars, okay? So nothing is sterilized. When you go for surgery, it's not sterilized in the surgical suite, but it reduces the risk to a level that is acceptable. And so in our case, we could recognize false positives. We don't mess up the science. So have we contaminated up there? Yes. We probably, with the right instruments, could find it. Whether it's still alive up there, if something went as a spore, and even the spore needs to come out in, so a Legionnaire spore or an anthrax spore will stay as a spore for a very, very long time, or a permafrost spore, until conditions are right. And so are conditions right on Mars? So yes, we've contaminated it, but it's within a reasonable level. We have all the information, and it doesn't seem to be, that wouldn't be the big risk.
Okay, I had two questions. One is real short. What's the risk of cross-contamination having happened already through meteors back and forth?
Okay.
And the other is that number, 300,000 spores, where'd that come from? It sounds like a huge number, and I was wondering how that was set as a limit.
It's a huge number, except for as microbes go, it's not a big number. I should have some, does anybody know what the number of microbes are on your thumb? Or, I mean, it's like billions in a handful of soil. So it sounds like a huge number, but it's not. And they're spores, not living organisms. And then the question about meteorites and natural influx. The scientists took that one on originally, too, because they're saying, well, heck, we've got stuff coming in all the time. And could things live inside of a rock? Yes. And they even did experiments on that. That being the case, then we should be able to detect some of it. Or maybe we are, as some people say, we're cousins to the Martians because life came from someplace else. We don't know. That's a good question. But just because a place had a forest fire before doesn't mean that you'd be smart to go throwing matches out the window as you drove through. So you still want to take care of it. And besides, when a meteorite is coming or an asteroid, those have been in space for thousands, millions of years. When we're moving things back and forth, we're bringing them back in a year. Big difference.
Thanks, Margaret. What were some of the back contamination mistakes of the Apollo mission?
There were some good ones. So when they first came in through re-entry, they opened the vents because the capsule, I guess, was hot. They opened the vents on the capsule. They landed in the ocean. And then the Navy divers jumped out of the helicopters and brought over the respiratory suits, respirators to the astronauts. So they were already right there. Then when they took the astronauts, so you notice they walked across the deck of the USS Hornet. So there's another place. Although they did have on biological isolation gear, BIGs. And those are on exhibit at the USS Hornet in Alameda if you ever want to go. And what was another one? Oh, yeah, they say that there were cockroaches running in and out of the lab at Houston. So it's nothing. I mean, so how do you seal up a facility? So there were many places where things could have gotten out. But again, the risk is not, that you don't reduce it to zero. You go to a lower level of risk. And we stopped it. We were convinced after Apollo 14 that there wasn't life on the moon and that it was okay. We were just protecting the samples, not the people after that.
I think this is a foolish question because I think I probably should have already heard the answer somewhere, but have we yet discovered any identifiable off-Earth microorganisms?
No, not to my knowledge. And I think that if we found it, you'd hear about it.
Could you comment on this and the work presented recently by this group in Scotland?
Oh, there are some groups who suggest, and there's a large number of people that are also talking about studying the aerobiome higher and higher. If you think about Venus, for instance, the habitat is not habitable down on the planet, and out in space it's not habitable. It's someplace in between. It may be warm enough, and they're also looking at aerobiome here. So what organisms can live up in space, and could they have their entire life cycle up there? So again, I'm always mindful of the distances involved. So when we talk about going up to Earth orbit, geostationary orbit, that's still pretty close. And when we're talking Mars and meteorites and asteroids, that's way, way far. So we do not have any evidence of that.
The Apollo astronauts brought back some hardware from the previous surveyor landers. And there's some controversy about whether Earth microorganisms were actually detected in that.
Yes.
What's your opinion on that?
It's not an opinion. NASA's Planetary Protection Officer, John Rummel, who is going to be affiliated with SETI Institute shortly. Oh, actually, he is already. Yes, he is now, yes. So he actually did a study of it going back. And as I showed you the picture of the Viking with all those guys and their skin showing it. So we didn't have the same kind of protections that we do in the lab. And so it's likely that the microbe that was found, the bacillus microbe inside of a camera, that was brought back was a lab contaminant. Not, so the best that they can tell, it was a lab contaminant, which says that it survived up there on the moon inside, inside of a camera for all those years. So again.
I wanted to ask about bringing back contamination by the sample returns mission that crashed like Stardust. I mean,
Okay,
that
that's
was
good. That's perfect.
Please talk about that in terms of planetary protection.
Yes, so there's one of those where it's the category. So remember I said there was 2 categories for a return, a restricted and an unrestricted? They flew through the tail of a comet. And they put up collectors that collected cometary dust and sealed it up and brought it back. Cometary dusts are not expected, so the National Research Council had done a study looking at what is a habitable environment. And you wouldn't expect that a comet, and especially the tail of a comet, would have conditions such that an organism could live there. Plus, when they collected it, they collected by impact on this, I forget, what's the name of the stuff? Aerogel. The aerogel, when it stops, it does a spike to 400 or 500 degrees C as it hits, and that's enough to kill anything that we know of. So again, it's life as we know it, but we do want to protect about that. So that mission, it actually turned out to be a success, even though it got really messed up when it came in. But the fact that it crashed and opened up was not a planetary protection concern. So it was unrestricted return. Same happened with Stardust as well.
Okay, thank you very much, Margaret, for these great talks on planetary protection.
Thank you.
As a token of our appreciation, I'm going to give you this amazing contaminated cup.
Okay.
Where you're going to be drinking contaminated coffee,
Excellent,
holding it
excellent.
with a contaminated hand.
You got it.
Around your contaminated colleagues.
Thank you very
Thanks.
much.